This is question 9 of the 2017 A2 Paper 1. It says figure 4 shows the stages of development of an insect called a damselfly. The adult damselfly uses a tracheal system for gas exchange. Explain 3 ways in which an insect's tracheal system is adapted for efficient gas exchange. In order to answer this question, students need to first identify a feature of the tracheal system and then go on to explain how that chosen feature allows for gas exchange. According to the examiner's report, many students could state that a short diffusion pathway is required, but they couldn't give the feature of the tracheal system that allowed this to make it an efficient system. Similarly, many students appreciated that a large surface area is required, but they could not sufficiently explain how this was achieved in an insect, i.e. with highly branched or numerous tracheoles. The basic pattern was to identify the feature and then explain it. Example, tracheoles have thin walls, so it's a short diffusion distance to cells. There are a large number of tracheoles, so a short diffusion distance. They are highly branched, so a large surface area. The trachea provide tubes full of air, so fast diffusion into insect tissues. There is fluid in the end of the tracheoles that moves out into tissues during exercise, so a larger surface area. And the last mark you could have had was to say that movement of insects using their muscles moves air in the tracheoles and so maintains a diffusion gradient. Lots of top tips for this question. In fact, the examiner's report wrote quite a lot. Basically, remember that spiracles are for water loss and are nothing to do with gas exchange. Remember to, when you're learning the material, construct a table that relates structure to function. And also, remember that the production of lactate causes water to be drawn out of the tracheoles, and that enables gas exchange to take place at a faster rate. Okay, so this question's quite straightforward, 9.2. The damselfly larva is a carnivore that actively hunts prey. It has gills to obtain oxygen from water. Some other species of insects have larvae that are similar size and shape to damselfly larvae and also live in water. These larvae do not actively hunt prey and do not have gills. Explain how the presence of gills adapts the damselfly to its way of life. So the key to answering this question was to recognise that it's asking you if an insect hunts, why it needs gills, and if an insect doesn't hunt, it doesn't need gills. And what the question is basically asking or testing the understanding of is a statement in the specification where it says students should be able to appreciate the relationship between surface area and volume ratio and metabolic rate, in other words, the demand for oxygen. If there's one part of this question that you need to have a go at, it's this one, because uncertainty is new to the specification. And again, like the log scale, it's going to come up again and again. So this question doesn't appear in any of the legacy material in any of the old specification. Let's look first at the uncertainty. Now, there I've labelled it the absolute uncertainty because what we're talking about is the minimum uncertainty provided by the instrument, basically. To make that a percentage uncertainty, what we need to do is divide the absolute uncertainty by the actual value and then times it by 100. So to work out our percentage uncertainties, we're going to do 0.19 divided by 1.61 times 100. And we're going to do 0.41 divided by 6.12 times 100, and that's going to give us 6.7. The question asks us to calculate the mean surface area of one side of one gill lamellae. Now, it's given us the measurements there, and so all we need to do to find the surface area is times 1.61 by 6.12. And that's going to give us our surface area of 9.85 millimetres squared. And they haven't given us the units here, so we need to make sure that we put the units in to get the mark. We also need to add together the two uncertainties from the two measurements that we had. So when we add 11.8 and 6.7, we get plus or minus 18.5. So you got all 3 marks if you got both answers correct. You got 2 marks if you got one answer correct, and you got 1 mark if some of the working there was correct. And my top tip for this question is to recognise that when they say uncertainty, plus or minus, basically they're not talking about the percentage uncertainty. And so to work out percentage uncertainty, we need to take that, divide it by the value and times it by 100. According to the examiner's report, only 7% of students were able to answer this question. So if you're aiming for one of those higher grades, make sure you've got this under your belt, and it'll be one of the things that sets you apart from the others and helps you to achieve those higher grades. Moving on to the next part of the question, a student used an optical microscope to observe part of a damselfly larva gill. Figure 5 shows the drawing the student produced. Okay, and so there we've got a cuticle and there we've got a hair. And the question asks, suggest 2 ways the student could improve the quality of her scientific drawing of this gill. Another fabulous question. So this question is basically asking you about practical skills. And there were many answers that you could have given. Number 1, you could have said don't use shading because that was used to represent the muscle. Number 2, you should have said could have said ensure lines are continuous and connected. As many places over the biological drawing, the lines aren't connected. You could have also said for marking point number 3, add further labels. However, it takes reading the examiner's report to realise that they don't just want you to say add further labels, they want you to give an example of the labels that you could have given. Adding further labels on its own was not credited with a mark. And this pushes the point that you can read the mark scheme, but you might get an inflated figure of what you would achieve because it takes years and years of interpretation and reading the examiner's report year on year to realise where the marks actually are. Don't cross-label lines is the 4th marking point, and also you could add to your diagram a magnification or a scale bar. Clearly a scale bar has to be calculated, but you see these often on many biological drawings. As always, I hope this video has been of use to you, especially the bit on uncertainties in biological drawing. What a fabulous question.